So today I'm actually going to focus on a research project we've been working on the 2 years leading up to the pandemic. And this project has applications in cancer therapy through the MAPK pathway. And I'll specifically discuss B-Raf and MEK kinase interactions and an allosteric site in B-Raf that we identify that is critical for the activity of this pathway. The MAPK pathway is initiated by growth factor binding to a receptor at the cell membrane, thereby activating a number of adapter molecules, which in turn activate RAS. Active RAS binds to GTP, which allows it to associate with RAF at the cell membrane. This causes a conformational change in RAF that frees this C-terminal domain from the N-terminal domain, as illustrated in this figure. Now, RAF generally exists in the cytosol as a quiescent RAF-MEK complex, and RAS binding actually promotes heterotetramer formation. This leads to activation, which leads to phosphorylation of MEK kinase, and then downstream activation of ERK kinase through phosphorylation events. ERK then targets different cytoplasmic and nuclear molecules involved in things such as proliferation, differentiation, and cell survival. Now, I should point out that RAF kinases have 3 distinct isoforms in humans: A-Raf, B-Raf, and C-Raf. The B-Raf isoform is the one I will focus on today, and it has the strongest activity towards MEK. But do note that these heterotetrameric complexes can form with other isoforms as well. Now, the B-Raf isoform has also been shown to be particularly susceptible to mutation, with over half of melanomas containing activating B-Raf mutations, of which over 90% involve a particular mutation, this V600E mutation. This mutation renders B-Raf constitutively active, resulting in unregulated activation of the MAPK pathway, which affects many hallmarks of cancer, such as proliferation, migration, protection from apoptosis, etc. And this activation is actually independent of both RAS activation and the formation of tetrameric complexes. And it does so by mimicking the negative charge of phosphorylation of the kinases activation loop, as illustrated here. These figures show 2 states of the B-Raf enzymatic kinase domain. This structure is bilobal. The smaller N lobe up here binds to ATP, while the larger C lobe down here binds to substrate proteins. The active site is found at the cleft between the 2 lobes. Now, in this state on the left-hand side, the A loop, which is colored in red, has residue 600. And so in the case of this V600E mutation, this glutamate is close to a glutamate at position 501 of this alpha C helix, which causes strong electrostatic repulsion. Now, in this state on the right-hand side, the glutamate at position 600 is near a positively charged lysine on the alpha C helix, which forms a favorable electrostatic attraction. This conformational change in the A loop actually releases its inhibitory interaction with the ATP binding site, which is found between the N and C lobes of the kinase. Thus, the V600E mutation favors this active conformation. Now, this here is just the kinase domain of B-Raf, but in wild-type B-Raf, the enzyme is also kept in the off state due to steric interference of its active site via this N-terminal domain, which consists of the conserved region 1 or CR1. Part of CR1 is made up by this RAS binding domain, which binds to RAS GTP. And binding of RAS GTP actually leads to a release of CR1 from the kinase domain, and therefore a release of kinase inhibition. And then in this case, the activation loop is then subject to phosphorylation and activation. But in the case of the V600E mutation, it leads to constitutive activation. Now, one thing not shown here is the fact that the release of inhibition also allows for dimerization, which is an essential step in the activation of many protein kinases, including the RAF family of kinases. Now, the fact that such a large percentage of melanomas is driven by one oncogene stimulated the search for B-Raf V600E inhibitors early on. The first B-Raf V600 mutant inhibitors were FDA approved for clinical use in 2011. Furthermore, drug discovery programs have largely focused on targeting the ATP binding pocket, but acquired resistance to B-Raf specific inhibitors is one of the greatest challenges for these targeted therapies. Additionally, acquired gain-of-function mutations in MEK have also been reported as a result of treatment with some drugs. Paradoxical activation through enhanced RAF dimerization in cells have also been observed, thereby showing the need for allosteric inhibitors and combination therapies. And in fact, combination therapies of MEK and B-Raf inhibitors have shown promise in the clinic. Now, we seek to explore alternative methods to inhibit the MAPK pathway that will help circumvent resistance, which is important in the field of drug discovery. Exploring alternative methods could also potentially offer insight on mechanisms behind the transactivation of MAPK pathways. Inhibitors that block protein-protein interactions are possible therapeutic targets in MAPK signaling, and in fact, some have already been pursued. For example, a small molecule drug had been discovered that binds to the RAS binding domain of RAF, thereby disrupting RAS-RAF interactions and in turn inhibiting downstream ERK activation. Now, recently in 2014, the crystal structure of the heterotetrameric B-Raf MEK complex was determined, and it's shown here. If you take a close look, you can see that B-Raf forms a conical dimer with itself, and each B-Raf molecule binds to MEK to form this heterotetramer. RAF-MEK kinases also sit face to face with their active sites and inhibitor pockets facing one another. Now, based on this structure, there are multiple sites one could potentially target to disrupt the heterotetramer. And it's already been established that mutation of this arginine 509 to a histidine, which is found at the center of the B-Raf dimer, can disrupt dimerization of B-Raf. Also, it's been determined that B-Raf MEK complex was found to have a dissociation constant of roughly 43 nanomolar, which indicates a very strong interaction. And so we hypothesized that the B-Raf MEK interface represents a potential target for inhibition of B-Raf activity and therefore of MAPK pathway inhibition, since this binding interaction may be critical for phosphorylation of MEK and the activation of downstream targets. And so we sought out to better understand regions that are critical for B-Raf MEK interactions specifically. The work I will tell you about has been performed largely by a talented group of undergraduate students taking a biochemistry lab course that I developed that is research-based, as well as collaborators at Penn. Before probing the B-Raf MEK complex, we first aimed to recapitulate complex formation using an E. coli construct of the kinase domain of B-Raf, so one in which the N-terminal regulatory domain is excluded. And we wanted to work with an E. coli version because yield, purity, and accessibility would make it more applicable for use in an undergraduate research lab, as well as for things such as high throughput screening. Now, many studies of B-Raf generally rely on the use of SF9 in sac cells for expression, since it's been very well established to produce the active enzyme. And that version was also used to determine the crystal structure of the RAF-MEK complex I just showed you. Now, the E. coli construct of the B-Raf kinase domain was generated by Sai et al., and it contains 16 point mutations found on the surface of B-Raf, particularly in the C lobe, and they are shown in green sticks here. These allow for expression and solubility in E. coli. Interestingly, however, this mutant was found to not be active despite no mutations to key catalytic residues. And this version of the mutant is still able to bind ATP. And even superposing this structure onto that of the wild type B-Raf, for example, from the B-Raf MEK complex, shows that they're structurally similar. So what's going on? We don't know. Well, using this construct one way or another would have been problematic because we needed an active form of B-Raf that could bind MEK. So we first tried to find a way to recapitulate its activity without losing the ability to express the protein in bacteria. So to try to figure out how to generate an active B-Raf, we analyzed these 16 mutations from the E. coli construct in the context of the B-Raf MEK complex structure shown here, but here I only show a dimer, not the full tetramer. Looking at the mutations, we found that there was one residue, particularly a phenylalanine at position 667, found on this alpha helix, that could make key hydrophobic interactions with MEK, which is shown as electrostatic surface here. Now, in the E. coli construct, this residue was mutated to a glutamate. And so this solubilizing mutation could have led to charge-charge repulsion with this acidic patch of MEK. And so our collaborator at Penn decided to revert the residue back to a phenylalanine found in the wild type. So that yields a construct now with 15 solubilizing mutations, as well as the activating B600E mutation. For simplicity, from here on out, I'm going to refer to this particular mutant as the B-Raf reversion mutant. And so we next expressed this construct in E. coli and tested its ability to bind and phosphorylate MEK. So for the rest of my talk, I'll discuss the characterizations, mostly biochemical nature, done on this mutant. So we first express our reversion mutant with a GST tag and used affinity purification before doing gel filtration. The size exclusion chromatography elution profile is shown here, and we saw that there were two peaks, both of which contain B-Raf, but the first peak contained aggregates. The second peak had minimal contamination of other proteins and has B-Raf. In fact, this B-Raf was found to be dimeric. And in fact, the V600E mutation has been shown to induce B-Raf dimerization in the absence of the N-terminal regulatory domain. In any case, we took fractions from the second peak for further analysis. To assess the ability of the reversion mutant, that is the mutant with 15 mutations and V600E, to bind MEK, and we compared it to the V600E mutant that is expressed in SF9 cells, one in which there aren't those extra 15 mutations. We performed pull downs. We also compared binding to wild type B-Raf as well as the original E. coli construct with 16 mutations and not the reversion mutation. What we found is that leaving the glutamate at position 667, so in the original construct, prevents complex formation with MEK, whereas the reversion back to phenylalanine restored B-Raf MEK complex formation, as shown here, and to a similar extent to what was observed in the corresponding SF9 construct. We next wanted to quantify their binding affinities. So to do this, we used biolayer interferometry, or BLI. We found that, consistent with the results from the pull downs, no binding with MEK was observed for the original E. coli construct with 16 mutations. But the reversion mutant could bind MEK, and it can do so with a KD of approximately 32 nanomolar. This binding affinity is approximately 13-fold higher than that of wild-type B-Raf, so the one that's not constitutively active, and on the same order of magnitude as the active V600E mutant that was produced from SF9 insect cells. Given that the B-Raf reversion mutant could bind MEK, and because different expression systems could generate proteins with distinct conformational, post-translational modifications, and therefore distinct activity levels, we next wanted to determine if this reversion mutant could phosphorylate MEK, its downstream target, and if it does so to the same extent and reaction kinetics as the B600E mutant produced in SF9 insect cells. To test this, we performed an in vitro phosphorylation assay to compare their activities, and we probed for MEK phosphorylation. This revealed a similar level of phosphorylated MEK by the V600E containing B-Rafs, that is, the ones produced in E. coli with or without a GST tag, compared to the one produced in SF9 insect cells. And these levels are higher than what was observed by wild-type B-Raf produced in insect cells, or the original E. coli construct with no activity. So we then used an ELISA-based assay for quantitative analysis of the kinase activity of these two B600E containing B-Raf constructs. We obtained Km MEK values for the E. coli construct and the SF9 construct of B-Raf, and found that they're both low nanomolar. This similarity is consistent with the Western blot data I show here, and with the KD values that were obtained using BLI. Thus, this illustrates that leaving in the glutamate at position 667 in the E. coli construct ablated complex formation between B-Raf and MEK, whereas the reversion mutation to phenylalanine allowed B-Raf MEK complex formation to occur. That key residue also restored activity in the E. coli construct, indicating tight complex formation is necessary for B-Raf's activity to phosphorylate MEK in vitro. Since reversal of the glutamate at position 667 to a phenylalanine was shown to restore the active state of RAF, suggested that this residue and the alpha helix on which it resides is important for RAF-MEK complex formation. To further verify the importance of this section of the alpha helix for complex formation, a student in my course, Linda, decided to mutate this phenylalanine to a tyrosine to see if it would enhance activity of B-Raf. And she reasoned that this mutation may introduce a hydrogen bond with a nearby aspartic acid at position 315 of MEK, or asparagine at position 319 of MEK, while preserving interactions with the MEK hydrophobic pocket. We tested binding by BLI and found that mutating that phenylalanine to tyrosine led to enhanced affinity to MEK by almost twofold due to reduced dissociation rate of MEK from B-Raf. And so this further confirmed the importance of this region of the alpha helix for complex formation. Now, the alpha helix on which that residue resides reaches into a tunnel in the MEK kinase, which again is shown as an electrostatic surface here, which seems to form a druggable pocket. And so we wanted to determine if the rest of this alpha helix, which spans residues 661 to 671, is critical for complex formation with MEK. To test this, students in my course analyzed the crystal structure of the complex and proposed mutations to test along this alpha helix to determine their effects on MEK binding. Specifically, they introduced the mutations that are indicated here based on potential interactions around these residues observed in the crystal structure, and their goal was to try to disrupt MEK binding. Specifically, we see here that N661 appears to hydrogen bond with arginine 234 of MEK, and mutation to a hydrophobic valine would eliminate this interaction. Mutation to aspartic acid, however, could either improve binding by the introduction of a salt bridge or maintain the same interaction as the asparagine. R662, shown here, contacts an acidic patch of MEK, and so its charge reversal to a glutamate would result in charge repulsion. Similarly, isoleucine at position 666 sits near a hydrophobic patch of MEK, and so introducing a charged arginine residue would lead to unfavorable interaction. Finally, this arginine 671 appears to hydrogen bond with the aspartic acid at position 315 of MEK, and so its mutation to an alanine would disrupt this interaction. As before, we tested binding by biolayer interferometry, and in agreement with our hypotheses, the N661B, R662E, and I666R mutants all had higher KDs than the original reversion mutant, which is shown here. The non-disruptive N661D mutation did not affect binding whatsoever, which is not surprising. Of note, the N661V and I66R mutations resulted in the greatest decrease in binding affinity when compared to the reversion mutant, and its binding affinity was decreased by more than tenfold. And this decrease was a result of both a slower association rate and an increased dissociation rate. Now, given that B-Raf must bind to MEK before phosphorylating it, we next wanted to determine if the mutations in the alpha helix, which span, again, residue 661 to 671, affect kinase activity. And so to do this, we use the same in vitro phosphorylation and ELISA-based assays as before. Of all the mutants tested, only the reversion mutants with the N661V and I66R mutations showed significantly reduced levels of MEK phosphorylation when compared to the reversion mutant shown here. And the I66R mutant in particular was completely dead. And this is consistent with the KM-MEK values we determined by ELISA, where the KM-MEK values of these mutants were found to be high, indeterminate in the case of the I66R mutant. The F667Y mutant, which again we generated to improve binding, showed a modest increase in activity in MEK phosphorylation under the conditions of this assay. Consistent with this, its KM-MEK value was found to be lower than the reversion mutant, which had a KM of about 144 nanomolar. Now, as expected, the original E. coli construct showed negligible activity, and MEK on its own also was not able to autophosphorylate. So any phosphorylation we see here was due to B-Raf. These data suggest that altering MEK binding by targeting that alpha helix can alter MEK phosphorylation levels. Since activated MEK goes on to phosphorylate ERK, we next wanted to determine the effects of our alpha helix mutant on ERK phosphorylation. So we used the same in vitro phosphorylation assay, but probed for ERK this time. Consistent with the levels of MEK phosphorylation observed, ERK phosphorylation was drastically reduced in reaction mixtures that have B-Raf reversion mutants with the N661V and I666R mutations, when compared to the reaction mixture in which the reversion mutant was used. This time, we also observed a reduction in ERK phosphorylation in reactions with the reversion R mutant with the R662E mutation, shown here. And it's likely that the conditions of this assay didn't allow us to observe a reduction in MEK phosphorylation, though its KM value was certainly higher than that of the reversion mutant. Now, of note, as before, unphosphorylated MEK could not phosphorylate ERK on its own, and ERK cannot autophosphorylate itself. So any phosphorylation we observed here was due to MEK being phosphorylated by B-Raf. These data, which are consistent with the dissociation constants we obtained from BLI, suggest that mutating the alpha helix can alter ERK phosphorylation levels by altering B-Raf affinity to MEK and thereby MEK phosphorylation. Now, from these data, we see that the I666R mutation appears to be the most effective at reducing B-Raf activity, because we could essentially not detect any MEK phosphorylation or ERK phosphorylation. And so we next wanted to determine if introducing this I666R mutation had an effect in cells in the context of a more biologically relevant B-Raf. That is, one in which the N-terminal domain is present, and one in which we did not include all these extra solubilizing mutations on the surface. So to test this, we introduced the I666R mutation into full-length wild-type B-Raf, in which we included the N-terminal domain, and we also included the activating V600E mutation. And we compared its activity to that of the wild-type B-Raf and the constitutively active B-Raf. So in other words, this mutant could essentially mimic a constitutively active B-Raf in cells that one has tried to inhibit by targeting just this one particular site, this I666 site with a small molecule, for example. And so we transfected HEK293 T cells with B-Raf and MEK, allowed it to express for 24 or 48 hours before subjecting the samples for Western to probe for MEK and ERK phosphorylation levels. Now, consistent with the in vitro data that I showed you on the previous slide, the mutant with the I666R mutation had lower activity, so lower phosphorylation of MEK, as well as ERK, when compared to the constitutively active version, but not as low as the wild-type B-Raf. And so the activity of this I666R mutant was likely higher because heterodimers with CRAF might still form in cells and activate MEK. So this suggests that maybe targeting the I666 site would be effective, but it may not be sufficient. And so combination therapies with ATP competitive inhibitors would be useful to further reduce B-Raf activity and prevent things such as resistance. So to summarize today, I talked about an E. coli construct that was generated by another group, but was inactive. And we basically recovered the activity by reverting one of the residues back. So our active E. coli construct had kinase activity similar to the constitutively active B-Raf that many labs produce in SF9 insect cells. Having a version that can be produced in E. coli would be quite amenable for high throughput screening programs for drug discovery, especially if one wants to find additional drugs against this kinase. We also showed that the alpha helix residue 661 to 667 is a promising target for drug discovery, especially because there are no current small molecules that have been shown to target this site. Through our cell-based studies, we also showed that targeting this alpha helix site alone is not sufficient, but combination therapies with ATP competitive inhibitors, for example, would be useful to further reduce deregulated B-Raf activity found in cancers and perhaps prevent the development of resistance. Now, there's still future work to be done. High throughput screens still need to be done to identify small molecules that target the B-Raf MEK interaction, and any hits from such a screen should actually be tested with ATP competitive inhibitors to look for synergy as well as effectiveness. One thing we didn't address in our study is the effect of targeting this alpha helix in CRAF MEK or a-Raf MEK complex formation. So we don't know how effective that is, and that's something that still needs to be looked into. It would also be of interest to figure out if targeting the RAF dimer in combination with the alpha helix and or ATP site could be effective. And some groups have started generating covalent inhibitors where they cross-link inhibitors against different sites of kinases to produce more effective inhibition. So it would be interesting to do something similar now with an inhibitor that targets this alpha helix region. And finally, there are other potential targets in this MAPK pathway one could potentially target. And one that we are looking at now is at the MEK complex interface. But there is no MEK complex currently or structure available. And so we're seeking to determine a three-dimensional structure before we try to find an allosteric site that could be targeted to inhibit MEK complex formation and therefore downstream activity. So with that, I'll end and thank everyone who's involved in this work. All my core students, and one of which who continued doing this work in my own research lab. Our collaborators at Penn, especially for their work doing the kinetic studies using the ELISA-based assay. Jihang Wang at the University of Sciences for providing us with the mammalian cell constructs of B-Raf and MEK, which we've used to generate our mutants and to do our cell-based studies, and funding from the college and the Pittsburgh Foundation for this work, and SBGrid, again, for the invitation to discuss this work, but also for their software support. A lot of the software, or some of the software that they support have been used by my students in the course, and having access to their support has been incredibly helpful to drive this research forward. And finally, I thank you all for your attention, and I'd be happy to take any questions.
Great. Thanks, Daniela. That was really interesting talk, and lots of work packed in there, and lots of students that are doing useful research stuff. So I guess we've already got a bunch of questions queuing up. If you have questions, please feel free to send them in chat, or people have figured out the hand raise option. So if you have a question and you'd like to unmute yourself, raise your hand and we'll go through those. So I guess to start off, there was a question about the affinity for the heterotetramer for B-Raf MEK, and if the heterotetramer affinity is weaker in the complex.
So the KD value was approximately 43 nanomolar. Oh, is the question, is the B-Raf MEK interaction weaker or stronger as a dimer versus a heterotetramer?
Actually, so Survey, would you like to unmute yourself and clarify?
Yeah, am I audible?
Yes, you are. Thank you.
Yeah, I was asking about the formation of the heterotetramer of the B-Raf MEK complex. So how does that compare in comparison to the B-Raf MEK dimer affinity?
That's a good question, and I'm not sure that has been determined. So I don't know what the KDs would be for the heterotetramer versus a dimer.
Okay.
But even, yeah, I'd have to check to see if they've at least tried it with the R509H B-Raf mutant, which doesn't dimerize, but I'd have to look further. I don't know offhand.
All right. Okay. Thank you.
Thanks. Another question about the I66R mutations and the others in the alpha helix is, so you tested them with the constitutively active background, in the constitutively active mutant background. Is it feasible to test them in the non-constitutively active?
Well, we could, except that we probably won't be detecting anything at that point because the wild type doesn't really bind or have any activity, or doesn't have much detectable activity. And the constitutively active one is the one we would want to target anyway with drugs, since that's the one that causes cancer. So that's the more relevant one to mutate.
So you couldn't do it, and if you could, there would be no point to it, basically.
Yeah.
So, yeah, thank you. so I guess kind of what may be a signaling naive question for me, but when you were mentioning that there was constitutive activity and you were thinking that that was because of some of the other CRAF or A-RAF variants that might be present, has investigating the MEK surface of the binding interactions for potential drug targets, is that something that you've considered and decided that B-Raf was a better target or the RAFs were a better target? Or is that just people have already looked at that hole and it's dry and there's no useful inhibitors there?
No one's looked, and I think actually the MEK hole would actually be more druggable because, you know, you can have small molecules that fit into that cavity. We just wanted to know if targeting that site would be important, and that's why we tested mutations on the alpha helix. But in fact, I think one would more likely find inhibitors against MEK rather than B-Raf, because the surface around that region of B-Raf is kind of flat. So it would be hard to find tightly binding small molecules.
Okay. Hindsight is 20/20, I guess. You know, if that you demonstrated the interface is important, and the immediate question is why didn't you look at the other side of it? So
yeah, it kind of accounts for both, but I think the MEK site or the MEK protein itself is more druggable in that region.
one last call for questions, and if not, then thank you again for presenting. That was a very interesting talk, even for somebody like me who's, you know, trying to catch up with signaling.
Well, thank you again for the opportunity.